Let me share the screen. Oops, shouldn't see that. That was an email. Good, that's done. Let's keep that. All right, let's go to this. Actually, I did want to say that I regraded a few things. Let me stop sharing the screen because I gotta bring the page back. All right, hopefully you're not seeing the screen. Let me see if I got that there. Yeah, you're not. Okay. I got a few things graded. I mentioned that in the lecture. I have the last lab, what the heck was that? Lab 5 graded, and you can make corrections by Saturday of this week for full credit. Anything else I need to talk about? I can't remember what else I wanted to talk about. I'll try and get the quizzes graded. Oh, I sent out the unknown number. So I did get that sent out late Tuesday night. And in a week, I will get you the first results from the first 3 tests. Now, the first time we have this contact, I will be emailing you everything. And what I'm going to do is just email the same file to everyone, and it'll have your number on it. Then you'll see the results for that number. After that, you will be requesting tests in your unknown notes page, which let me see if I can show you that. Are you seeing my screen? Are you guys seeing the screen?
No, just you.
All right. So you should put your unknown number in your unknown notes page, which is right here. And you should also put your results when I send them to you on Tuesday. So you don't need to do anything until Tuesday, but I will need your unknown number, and you will need to put your results, which are observations, and your conclusions in the unknown notes page. You should also list out the bacteria, which will be under consideration, and then state in your unknown notes page, usually at the bottom of the page, what test you want to run. And you can take a look at the unknown list for figuring out what species you might have and what test you'd like to run. If you have any questions about the unknown project, please ask, because some people just let it sit and then get confused and then never ask and then never turn in the unknown project. It's worth 75 points, and that will be enough to lower your grade 1 grade. Okay? Any question about any of that? So if you have any questions about the unknown project, ask. And you can, in that one, you can email me, but you can also ask in the unknown notes page. I will check the unknown notes pages starting on Tuesday, every day, Monday through Friday. I probably will not check them on Saturday and Sunday. I get a break too, but I will check them on Monday. And that could be a problem if late on Friday you ask for a test. You're not going to have me see that until Monday. So understand, late on Friday, you might not get any results until Monday or later, depending on how long the test has to incubate. So be careful of that. And I should check those pages about every day, Monday through Friday. But I'm not going to guarantee I'm going to check them. I'm probably not going to check them on Saturday and Sunday. Any questions about any of that? All right, if not, let's get off of that.
Just to clarify, so the first 3 tests, you said we don't have to ask for those. You'll give those to us automatically, and then going forward, we'll have to ask for the specific tests we want, right?
Correct.
Okay.
Yeah. I'm giving you the first 3 tests because I can then reduce the number of emails by 60. Well, 59, because I'm going to send 1 email out to everybody. But that really makes this project doable, because if it's over 100 contacts, I guess not emails, but contacts through the unknown notes pages, that's just too many for one project. But 40 is a doable number. And so I'm giving you the first 3 tests, and you don't get to choose these. And they're actually good 3 tests. Okay, meaning I did go through and pick out good ones. Gelatin I picked out because it'll take you a week before you'll get the results. And you need to do the gram stain for sure. And then the lactose fermentation is a good test. So those are all 3 good tests. All right, so we're doing lab 6 today. Let's go to lab 6. So there's some pre-reading that you should do before doing the lab, and you can take a look through that. This has a little more reading than normal, but do the reading. There will be some video clips, and I think there's a virtual lab today. I'll have to, let me open up the newer version to see if there's a virtual lab today. I think there is a virtual lab today. Yep, there's a virtual lab today. The teacher's edition, I just never have updated it. And since it had everything marked of what I wanted to say, I kept the older teacher's edition. You won't see the teacher's edition. Upon successful completion of this lab, you should be able to identify the type of chemical agent and the active ingredient used in the various disinfectants and our antiseptics, be able to evaluate the effectiveness of a disinfectant or antiseptic using the diffusion method, be able to determine the mode of action of various antibiotics. We'll look at antibiotics and talk about them today, and they have a different mode of action. Evaluate the effectiveness of various antibiotics using the diffusion method. This will be mainly what we're doing in this lab today. Item number 4. A list of different variables that influence antibiotic diffusion susceptibility testing, and then finally be able to define the terms with sterilization, disinfection, a disinfectant, an antiseptic, a bactericidal agent, a bacteriostatic agent, what's an antibiotic, what's a spread plate, and what's the zone of inhibition. Any question about what we're doing? All right. So sterilization means that you're eliminating all viable organisms, including endospores. In the lab, we normally would give you sterilized media that has been sterilized by putting it in an autoclave. And if you don't know what an autoclave is, it's a giant pressure cooker, and it both heats and puts things under pressure to get them sterilized. And that's one easy way to kill endospores. As you know, endospores are difficult to kill, and we'll talk a little bit more about that later in the term. There are a few things that can kill endospores, and an autoclave is an easy one. But obviously, you cannot put a patient in the autoclave and then kill off the bacterial infection that has endospores, because the autoclave would kill the patient. Disinfection refers to the destruction of vegetative cells, not necessarily endospores. A disinfectant is a chemical agent applied to an inanimate object. Usually, a disinfectant is pretty harsh it would not be something you would necessarily want to put on your skin. Like when you clean the toilet, you use a disinfectant. An antiseptic is something that's a chemical agent applied to living tissue. And that would be something like iodine. We use that as an antiseptic, and it's not harmful to our tissue, but our tissues, but it is harmful to bacteria cells, for example. Okay, any question about any of that? To evaluate the effectiveness of a chemical agent, the surface of a nutrient agar plate is inoculated with a particular bacterium. And usually we have the bacteria growing everywhere, so we say it's a lawn of bacteria. Excuse me here, I'm going to sneeze, I think. If I blow my nose, I won't sneeze. Hopefully that'll do it. I didn't really blow my nose, but I tried to. I still have a feeling like I'm going to sneeze, but it's a little less intense now. Where was I? Oh, you're growing the bacteria on the disc, and the bacteria is everywhere. So we call it Elanobacteria. It will grow with so many things. You won't see individual colonies. You would just see bacteria everywhere on the disc. The chemical agent is then put down on the surface of the agar before the bacteria is incubated. The chemical agent then diffuses into the agar medium, establishing a concentration gradient around the disc that contains the agent. You then incubate the plate, usually overnight, and the bacteria grow on the disc. If there's a zone of inhibition around the disc, that's an area of no bacterial growth, then the chemical agent is assumed to be effective for that particular bacterium. Conversely, if there's no zone of inhibition, then the chemical agent is assumed to be ineffective. And that's a simple way of doing it. This table is showing you the different chemical agents and their mode of action. Where the heck is that? Well, the general action, how they're killing, it's right here. How we use them, like ethanol and isopropanol, is used as an antiseptic. It's a very weak antiseptic agent, so usually we call that a degerming agent. Formaldehyde, formalin, glutaraldehyde, a disinfectant, iodine, an antiseptic. It can be used as disinfectant if you put the iodine, or the chlorine is used as a disinfectant. Sorry, chlorine bleach, for example, is used as a disinfectant. Anyways, that table tells you the different chemical agents and how they're used and their mode of action. The zone of inhibition size depends only partially on the sensitivity of the organism to the disinfectant or the antiseptic. There are various other factors that affect the zone of inhibition, including the concentration of the chemical in the paper disc, the molecular weight of the agent. The larger the molecular agent is, the less it will diffuse, or the distance will be less it will diffuse in the agar plate. And then the density of the bacterium can also affect the size of the zone of inhibition. The more dense and the older the bacteria are, the smaller the zone of inhibition. Okay, so those are a few of the agents that can, or factors that can affect the size of the zone of inhibition. To evaluate the antibiotic effectiveness, we obviously here are using antibiotic used as a bactericidal agent if it kills the bacteria, or as a bacteriostatic agent if it simply prevents the bacterial growth. Generally, bactericidal agents are preferred, but bacteriostatic agents can be used, and they do not kill the bacteria, they just stop them from growing. And then we're relying on the immune system of the patient to assist in elimination of the infection. Any question about any of that? When you look at a plate, you cannot tell if the agent is bactericidal or bacteriostatic just by looking. You have to do a test to determine if the agent is bactericidal or bacteriostatic. And that is one of the questions for you in the worksheet. The Kirby-Bayer distillation test is the most common test used for testing for antibiotic susceptibility. The results of such testing help a physician in choosing which antibiotic to use when treating a sick patient. Bacteria are isolated, grown in a pure culture, and then put on the plate where you say that they can grow everywhere. Let me blow this up so you can see that better. You can see this is what the agar plate looked like before putting on the bacteria. And then we put bacteria on so that it grew up kind of a whitish color, practically everywhere, except in the zone of inhibitions around the disc. And that we call a lawn of bacteria. You then, before the bacteria are grown, just a few minutes after you put down the bacteria, you then put down the disc containing the antibiotic. This will then form a gradient of antibiotic around the paper disc. And as you go further out from the paper disc, you'll have less antibiotic. You'll have a zone of inhibition wherever the antibiotic concentration is great enough to prevent the growth of the bacteria. Any question about any of that? All right, so we'll talk a little bit more about how you do this in the lab, but you then incubate the plate with the disc and the bacteria and allow the bacteria to grow. And then you bring the plate out and you take a look at it. And you notice which have a zone of inhibition, where the antibiotic concentration is great enough to prevent the bacteria from growing. The size is dependent on several factors. It is dependent on the rate of diffusion of the antibiotic. A smaller antibiotic will have a larger zone of inhibition than a greater or larger antibiotic. So that will affect the size of the zone of inhibition. The sensitivity of the microorganism will also affect the size of the zone of inhibition. One that has a larger zone of inhibition may have a greater sensitivity to that antibiotic than a bacteria which has a smaller zone of inhibition. The growth rate of the bacterium will determine the size of the zone of inhibition. And there are a number of variables which can affect the Kirby-Bayer distillation test. So when we do the Kirby-Bayer distillation test, they're always standardized so that when one person does the Kirby-Bayer test, no matter where they are in the world, no matter what the time of day it is, they will get similar results because the Kirby-Bayer test is standardized. You always, when you're doing the Kirby-Bayer test, you start with bacteria that come from several bacterial colonies that's been grown on non-selective media. and it's been grown for less than 24 hours old. So you want young bacteria because it's quickly growing, and young bacteria is also sensitive to, more sensitive to the antibiotic than older bacteria. You then grow it Well, you then grow it for less than 24 hours. I think you grow it from something like 15 to 18 hours, but I don't remember. And it's not saying here, it just says less than 24 hours. And then you take the liquid culture and adjust the broth turbidity, how many cells are in it, to 0.5 McFarland units. And that will be approximately 10 to the fifth organisms per mil. So this is just an easy way to make sure that everyone is putting the same number of bacteria down on the disc, and that the bacteria is grown for about the same amount of time. You then inoculate at room temperature, always on a Mueller-Hinton agar plate. And the pH of that agar plate, Mueller-Hinton is a type of media. The pH of that is between pH 7.2 and 7.4, meaning the pH can affect the zone of inhibition, the size of the zone of inhibition. The media can affect the size of the zone of inhibition. The agar plate is always going to be 4 millimeters thick. Different size of the agar, different depth of the agar will affect the diffusion of the antibiotic. So you always use Mueller-Hinton plates, 4 millimeters thick. The diameter of the plate should be 150 millimeters. And then you spread the bacteria on the plate using a non-toxic swab, and you do it so the bacteria will grow everywhere as a confluent growth. We call that a lawn of bacteria. You then allow the disc to sit for about 5 minutes. That's to absorb the liquid from the bacteria, and then place the disc on 24 millimeters apart, no more than 12 discs in the plate. You add the antibiotic disc 5 minutes, but no longer than 15 minutes after adding the inoculum of the bacteria. You then put the disc in a non-CO2 incubator and incubate for 16 to 18 hours, growing the bacteria at 35 degrees Celsius. And you always put the plates in between 0 minutes to 15 minutes after adding the antibiotic disc. Any question about any of that? So that's highly standardized, so that when somebody in one part of the world is doing Kirby-Bayer test on the same organism with the same antibiotic, they will get a similar zone of inhibition as somebody else running the Kirby-Bayer test someplace else in the world. After incubation, you then take a look at the inhibition zones, the size of the inhibition zone, and then you measure the size of the zone using millimeters. You then consult a chart set up by the National Committee for Clinical Laboratory Studies, which will then tell you whether the bacteria zone of inhibition is a bacteria that's sensitive to the antibiotic or resistant to that antibiotic, or something in between that we call intermediate. And that will be dependent simply on the antibiotic choice and the size of the zone of inhibition. For example, with Staphylococcus aureus, if we were to look at the chart, if the size of the zone of inhibition is 20 millimeters or less, for penicillin, then we say that Staphylococcus aureus is resistant to penicillin. If it is greater than or equal to 29 millimeters, this size of the zone of inhibition, we then say the Staphylococcus aureus is sensitive to penicillin, and this is penicillin G, by the way. If it's in between 20 millimeters and 29 millimeters, we say that the size of the zone of inhibition is intermediate for Staphylococcus aureus to penicillin. Any question about any of that? And this is showing you how you do the measurement. You actually measure the diameter in millimeters from one end to the other. And usually you take two measurements and then take the average of that measurement. So this one here, what would that be? Resistant, intermediate, or sensitive to penicillin? And we're going to assume this is Staphylococcus aureus.
Sensitive.
Anyone else want to guess? Oh, there's the measurement there. Okay, what's that measure? It's less than 29, so it's not sensitive.
Would it be resistant?
It's resistant. It looks like it's 19 or 20. I don't have my glasses. Let me magnify that. Let me blow it up a little bit and then I'll be certain. But it's definitely 20 or less. It does look like it's 20, which means it is equal to 20, so it is resistant. Okay. All right, you should know the terms of this lab. You're not going to be physically performing this lab. Everything's going to be done online, but you will be responsible for knowing how to interpret the effectiveness of antibiotics using the Kirby-Bayer diffusion test. And what we'll do is we'll give you a picture and there will be a ruler on it and you've got to measure it. And not really a ruler, it'll be a caliper. You have to measure it and then make your interpretation from the table what the zone of inhibition is. Is it resistant, intermediate, or sensitive? For this lab, there's a video clip to watch on preparing how to make a spread plate, and then review the steps for using aseptic technique for preparing a spread plate or a bacterial lawn or growth plate, and that's detailed here. You then inoculate as much of the agar surface as possible using a swab, including inoculating the edges. And let's go ahead and look at a plate here. I don't have a plate here. There we go. So to inoculate this plate, what you do is you get a cotton swab. We're going to pretend this is a cotton swab and this is the cotton edge. Can you guys see that? Yeah, you can if I bring it over my face. And what you do for the plate is you inoculate everywhere on the plate. It's going back everywhere, making sure you get everywhere. And then you rotate the swab a quarter of a turn and rotate the plate a quarter of a turn, and then inoculate a second time, going everywhere. Okay? And then you inoculate the swab or rotate it a quarter of a turn. And I can't show that with my hand, so I'm just going to do it again. A quarter of a turn, and we did a quarter of a turn, and then inoculate the plate everywhere with a cotton swab. And then what you do is you take the plate and then you take the cotton swab and you go around the edge of the diameter of the plate. Okay, so you go around the edge of the plate. I guess I can show it with the mouse right here. And you have to make sure you have the bacteria everywhere. I once had a student, what she did was she went like this, and then rotated the plate, and then rotated the plate. And then she had these large gaps everywhere on the plate, and she couldn't measure the zone of inhibition. And I looked at that and I said, well, you don't have a line of bacteria. You don't have the bacteria everywhere. When you streaked out the bacteria on the plate, you were supposed to streak it everywhere. So I don't know, you do the cotton swab, something like 20 to 30 times everywhere across the plate. So the bacteria is growing everywhere. And why you're doing it 3 times is to make sure that bacteria will grow everywhere. Any question about any of that? Now, where was I? All right. And then use words to explain the spreading pattern you're going to get, and that is you're going to get a lawn of bacteria growing, assuming you don't have the antibiotic disc on it. All right, this doesn't have the antibiotic effectiveness. For evaluating the antibiotic effectiveness, you then have a video clip on preparing a culture for the Kirby-Bard disc diffusion test, and then a video clip on interpretation of the results for the Kirby-Bard disc diffusion. Then you have a virtual lab, which this program will download and will take some time to load on your computer. But once you get it, it should be able to show you how to do this procedure and how to measure the evaluation of the antibiotic effectiveness. Just click the links for the description, the steps, and the restart button. You do need to restart 3 times to see each of the 3 plates. And let me see if I can get that to get started. Go in on it. All right. So I'm going to continue talking about this and remind me to come back to that. Well, oh, sorry. This is the older one. I got to go to the new lab here. Oh, I already have it open. That's good. There we go. Oh, that opened faster than I thought it would. All right. So you can read about the description, the steps you do. Click the start to get started. And here we're seeing a plate showing you E. coli. And there's different antibiotic discs. The TM stands for something. And you look up here, you know, I don't know how you say that one, tropomyosin, TM. And you need to take the ruler and measure the diameter of that disc, and then come up here, and this chart tells you if it's less than 12 millimeters, it is resistant. Boy, that's not showing you. And if it's greater than 15 millimeters, it's sensitive. And then you, oh, there we go. For that one, it's really easy. It's 0. For this one, you bring it right there and move it there and measure that, and that looks like about 4 centimeters, so that would be 40 millimeters. And then for SIP, I don't see SIP here. Does anyone see SIP? Oh, there it is. So that would be sensitive. And then you mark it there, and that'll be your selection, the green one. And then when you get all this done, you can click something and it'll tell you which answers were correct and which are wrong. And you can redo it again to get everything correct if you want, although that's not required. All right, any question about that virtual lab? You'll actually be able to do this in the lab. Remember, you got to restart it 3 times because there's 3 different plates. There's the E. coli. There's Probably Pseudomonas aeruginosa. I can't read that very well. And then Staphylococcus aureus. Any question about that? All right. So using the standards required for the Kirby-Bauer susceptibility testing on E. coli, we have a spread plate technique. And antibiotic discs are placed on the surface of the agar and incubated overnight at 35 degrees. Below are the results along with the NCC-LS zone table showing you the standards that are used for the interpretation of the zone of inhibition. So this is the agar plate, and there's the caliper for measuring the size of the zone of inhibition. Now, when you use the caliper, you don't start from this side to this side of the caliper because the 0 doesn't start here. So this is not the measurement of the zone of inhibition. The measurement of the zone of inhibition is the 0. Everyone got that? So for ampicillin, I'll give you the measurement. And that is for ampicillin on E. coli, the zone of inhibition is 15 millimeters. And that would be right here for ampicillin. So use this for ampicillin, cephalophyllin, gentamicin, and tetracycline. The chart you're going to use is right here, and we're only giving you results to E. coli. And then put your answer for the size of the zone of inhibition in this table. You can actually add the size right here, right in the table. And then the interpretation. Tell me, is it sensitive to the antibiotic, intermediate to the antibiotic, or resistant? You just need to type S, I, or R for that. Okay, so that's the first question to answer, which I think is question 2. Yep, question 2. And we should look at the worksheet, because I might have redid the numbering. 2 and 3. Yeah, I did redo the numbering. So the next question will be number 4. In question number 3, in 2, you fill in this table. In 3, you list the 5 variables that must be standardized when performing antibiotic susceptibility testing using the Kirby-Bauer method. And you have to list 5 for full credit. And then question 4, the size of the zone of inhibition often varies between two different species of bacteria when challenged with the same antibiotic. Answer, what is a possible explanation? 5, what would happen if we didn't use aseptic technique to put the antibiotic disc on the plate? 6, what would happen if we increase or decrease the concentration of the antibiotic in the disc? Be specific in your answer. And you do have to answer it for both increase the concentration of the antibiotic and decrease the concentration of the antibiotic. 7, antibiotics can be bactericidal, that is, they kill the microbe, or bacteriostatic, they inhibit the microbe, but do not kill it. When observing a zone of inhibition around the disc, how would you determine whether the antibiotic was bactericidal or bacteriostatic? 8, when performing antibiotic sensitivity testing, it's important that the test conditions are kept very standardized. Why? Answer why. 9, are there other substances besides antibiotics that we could evaluate using this method, the disc diffusion method, the Kirby-Bauer disc diffusion method? 10, lastly, does the presence of the zone of inhibition necessarily mean the bacterium being tested is susceptible to the antibiotic? Explain your answer. All right. We're now done with the lab. Go ahead and start working on the worksheet. I will be here until 8:30 to answer any questions. But when the last student logs off, I'm going to log off as well. So make sure you ask your question before 8:30. All right?